Inviting the next speaker, I have a couple of announcements. This INCD conferences we organize under the umbrella of Society for Nutraceuticals and Chronic Diseases. So this society started 6 years ago, and we have organized, this is the 5th conference we are organizing. So I would like to encourage everybody to take membership of this society. And the first thing. Second, there is a panel discussion today in this hall. After the panel discussion, we will be organizing the general body meeting of the society. So please attend this general body meeting. Thank you. Our next speaker is Professor IL26 Mandal, and he is from Department of Biochemistry, Central University of Rajasthan, Ajmer. And his areas of interest are understanding molecular mechanisms of dysregulated gene expressions and cellular signaling network associated with the cancer, exploring the role of various metabolic diseases including diabetes, obesity and hypercholesterolemia, environmental risk factor on the peculiar differentiation property of epithelial cancer cells. He received his PhD from IIT Kanpur in biochemistry in 2005, and he did postdoctoral research at the University of Texas Health Science Center at San Antonio. And he is the editorial member of various journals like Scientific Reports, Plus One, BMC Cancer, Frontiers in Endocrinology, etc. So I would like to invite him for his talk.
So there is one announcement. We are going to have another group photograph. We had one yesterday, but there are many new people who have joined, and it will be a totally different group. So we can actually gather around after the panel discussion is over. So we'll gather around in the same foyer where we are actually having a INCD and Deepak and Amoli is there. Please gather there. Just immediately after the panel discussion, you all are welcome to be there. Thank you very much.
Also talk about the cultural evening.
Yeah, and there's another next announcement is, if anybody who is not aware, we have our cultural evening and dinner in this premises only, and there's no need to go to the zoology department. It's totally wet, and it was outdoor event, but now we are going to do it indoor. Thank you.
A very good afternoon to all of you. Before I start, I would like to give a special thank to organizing committee, especially to Professor Alak Chandra Bharati and our session moderator, Professor Gopal Kundu and Professor Ajay Kumar, and the other audience for over here. Today I will give a talk on the title, A New Drug Targets Dysregulated Cholesterol Metabolism for Prevention of Cancer Growth and Metastasis. So, before I start, I would like to just give a brief introduction. I know everyone is known about this still. There are many factors, especially the environmental factors, usually the pollution, toxin exposure, meat consumption, alcohol consumption, physical inactivity, smoking. These are the environmental factors which always modulate the cancer potential, cancer development, cancer metastasis. In parallel, this environmental factor can also modulate different body parameters, as for example body weight, obesity, cholesterol, diabetes, family history. So these are the intrinsic parameters which can also directly or indirectly involved in cancer progression and metastasis. So today I will be discussing how the important role of high cholesterol in cancer progression and metastasis. So in the beginning, in 2015, when I have started my independent career in Central University of Rajasthan, so I have started the work that the top 50 countries which have highest cancer mortality rate, I have just pointed in the world map, and we have noticed that the top 50 countries, they are situated in a particular zone. Similarly, when we have marked the least 50 countries which have lowest cancer mortality rate, their position is in different. So you can see that highest mortality rate country, they are situated from the Tropic of Cancer towards the North Pole or Tropic of Capricorn towards the South Pole. Whereas the country which have lowest cancer mortality rate, they are situated in between Tropic of Cancer and Tropic of Capricorn. Basically, they are nearby equatorial zone. So they have somehow geographically distinct. So later on, I have again marked the top 50 countries which have the highest average cholesterol level in their population, and you see that they are the top 50 countries which have highest average cholesterol. They are situated from Tropic of Cancer towards the North Pole and Tropic of Capricorn towards the South Pole. So this geographical situation actually mimics to the geographical situation of the top 50 countries having highest cancer mortality rate. Similarly, you can see over here the least 50 countries having lowest average cholesterol. They are situated in between Tropic of Cancer and Tropic of Capricorn. It is almost similar to the country which have lowest cancer mortality rate. So from this data, you can guess that there could be a link between the cholesterol and cancer mortality. So in that way, we have started our research career as independent. So then we have read many papers and we have noticed that yes, the high cholesterol rich diet shows an increased risk of various cancer including pancreatic cancer, colon cancer. Elevated serum cholesterol or LDL cholesterol may increase the risk of several cancers including breast cancer. Even cancer cells watch their cholesterol level to increase their growth. So in this context, we have started our research and we have compared the cancer mortality rate and cholesterol level through statistical analysis, and we have noticed that the average cholesterol of a country is directly proportional to the cancer mortality rate of a country. So this is the correlation coefficient, and then we have again counted the cancer mortality rate of different anatomically specific cancer versus average total cholesterol, and we have noticed that for various type of cancer like lung cancer, bladder cancer, ovarian cancer, pancreas cancer, breast cancer, there are positive correlation between average cholesterol and the cancer mortality rate. However, we have not seen the positive correlation in case of liver and cervical cancer, rather it is a negative correlation. So then we started the research in clinical sample analysis, and we have noticed that the patient of cancer blood have high cholesterol level as compared to the patient of healthy blood. Similarly, the cancer tissue contain elevated cholesterol level as compared to the benign tissue. This is the breast cancer tissue. And then we have compared the cholesterol level between the low metastatic MCSF7 breast cancer cell and the high metastatic breast cancer MDMB231 cells, and we have noticed that highly metastatic MDMB231 cells have the more cholesterol as compared to the low metastatic breast cancer MCSF7 cell. So this data indicated that somehow cholesterol is associated with the cancer progression. It can be also associated with the metastasis of the cancer. So then we have analyzed the cancer sample of breast cancer, malignant breast cancer, as well as the benign breast cancer, benign breast tissue, and we have checked the different cholesterol regulatory genes like HMG CoA reductase. It's a rate-limiting enzyme in cholesterol biosynthesis pathway. LDLR, it's a low-density lipoprotein receptor. It is also very important for cholesterol internalization from outside to inside the cells, and the SRBF1, it's a crucial transcription factor which regulates the cholesterol biosynthesis. So we have noticed that all these crucial cholesterol regulated markers are more in the malignant breast cancer tissue as compared to the benign breast tissue. Similarly, we compare the cholesterol regulatory genes between high metastatic MDMB231 cells and the low metastatic MCHF7 cells, and we have noticed that the high metastatic MDMB231 cells have higher level of HMG coreductase, LDLR, and SRBF1. So this data further showed that not the cholesterol is more in the cancer cells, even cholesterol regulatory pathway is more active in the cancer cells. Then we have again charts various genes, those are related to cholesterol pathway, and in that time we have noticed that there are a gene set that is RXRA, SO81, squalene epoxidase, and ACAT1. These are the 4 genes. Out of these 4 genes, 2 genes are always upregulated in the cancer and 2 genes are always downregulated in the cancer. We have analyzed this data through TCGA database, and we have made the survival curve by combining all 4 genes together, and we have noticed that when we combine 4 genes together, the survival curve is poor for all cancer. We have mentioned over here bladder cancer, breast cancer, head and neck cancer, kidney, renal cell cancer, liver cancer, as well as the lung cancer. So these data further suggested that these 4 genes could be a gene signature when the cholesterol associated tumor is there. So to validate this thing, we have analyzed the tumor samples and between actually the control and tumor, and then we noticed that the tumor, this is best tumor samples, and we noticed that the best tumor tissue have higher level of SOT1 and squalene epoxidase, whereas the RXCR and ACAT1 is downregulated. So this experimental data satisfied that the 4 genes could be the gene signature when the cholesterol associated cancers is there. So then we wanted to know whether this cholesterol pathway can block or not. So everybody knows that there are many drugs which can block the cholesterol pathway. Especially I have worked simvastatin, and we have already proved in animal model that simvastatin can prevent the cancer growth and metastasis in a new mouse model. But today I will be discussing another new drug, that is ADA. It's actually the conjugation of dopamine and arachidonic acids. Dopamine and arachidonic acids, sometimes they conjugate in our body, that is, our name is NADA, N-acyl dopamine, and there are few papers that NADA can be can show the anti-cancer potential. Actually, this is the work in collaboration with Russian group, and also one of the collaboration is Professor Gopal Kundu is over here. And so we have shown that this ADA can show the anti-cancer activity as well as the anti-migration EMT activity, and you can see over here some of the data I am mentioning over here that ADA can prevent the cell migration, it can prevent the colony formation, it can also prevent the epithelial to mesenchymal transition, as well as that can prevent the soft agar formation. So ADA can prevent cell migration, stemness, as well as the epithelial to mesenchymal transition. And then we have to know whether because there are few reports that there is a link between ADA and cholesterol, then we thought that whether the ADA can block the cholesterol level in cancer cells or it can decrease the, so we have treated the cancer cells with ADA, and we have found that the cellular cholesterol level of MDMB231 cells is decreased when cells were treated with ADA. And then we have treated the cells with ADA and ADA plus cholesterol. We have found that ADA inhibited cell migration, stemness property was increased when we have treated with ADA plus cholesterol. Means cholesterol treatment reversed back the ADA mediated inhibition of cell migration, colony formation, as well as the matrix metalloprotease activity. Then we have to know whether the ADA can prevent the cholesterol regulatory pathway. So we found that ADA treatment can inhibit the HMG coreductase, LDLR and SRBP1, and also it can inhibit the SRBP1 protein level. So ADA can prevent the cholesterol pathway to decrease the cholesterol content in the breast cancer cells. So then we have to know whether the ADA can prevent the breast cancer induced bone metastasis. So in culture model, what we have done, we have used one osteoclast precursor cells, that is called raw 264.1 cells. That cells can do the osteoclast activity after a certain condition. So when we added the conditioned medium of breast cancer cells, we have noticed that the osteoclast activity is more, that is measured by TRAP activity. The TRAP activity is a traditional method to measure the osteoclast activity. Then we measure the TRAP staining of osteoclast cells. It can show the multinucleated osteoclast cells, and we notice that breast cancer conditioned medium can increase the TRAP activity of osteoclast precursor cells. So this is the indication that the breast cancer can increase the osteolytic metastasis, and to further verify, we measure some osteoclastic marker activity like the TRAP, cathepsin, and IL26 is the master regulator of osteoclast activity. We have noticed that conditioned medium can increase the TRAP, cathepsin, and the master regulator transcription factor IL26. So then we thought whether the ADA can block the breast cancer induced osteoclast activity. We have noticed that the ADA can decrease the breast cancer induced TRAP activity, and there are two crucial osteoclastic factors, that is called CSF1 and RANK ligand. One is cholesterol factor, another is receptor activated NF-kappa ligand. These are the two crucial factors which can activate the osteoclast activity. These are also highly expressed in the cancer cells. So we have noticed that whether ADA can block the CSF1 and RANK ligand level in breast cancer cells, and our data suggested that ADA can inhibit the CSF1 and RANK ligand in breast cancer cells. So further to verify this thing, we have analyzed some tumor samples, and we have noticed that malignant breast tissue has a high level of CSF1 and RANK ligand level as compared to the benign breast tissue. So these data suggested that the CSF RANK ligand is more in the metastatic breast cancer, and ADA can prevent the CSF1 RANK ligand activity to block the breast cancer induced bone metastasis. So this is a cartoon. You can see that the SRBP1 is a crucial transcription factor which can regulate the cholesterol level, and this cholesterol can increase the CSF1 and RANK ligand, and this CSF1 and RANK ligand actually can increase the IL26 level in osteoclast precursor cells. This IL26 is crucial for the osteoclast activity by releasing cathepsin K and matrix metalloprotease. And in this case, ADA can prevent the SRBP1, which can decrease the cholesterol level. As a result, breast cancer induced osteoclast activity can be inhibited by the treatment of ADA. So this is the first report that ADA can inhibit the cholesterol level, and it can also inhibit the breast cancer induced osteoclast activity in cell culture model. So before I ending, I would like to say about my collaborator, the Professor B. Benjugulov. He is from Russia. Actually, this is the Indo-Russia project, and he is the collaborator. He has given the compound ADA. Professor James Nadesovich from University of Illinois, Professor Alona, and Professor Gopal Kundu, and Dr. Logendra Sharma from Sanjay Gandhi Postgraduate Institute of Medical Science, and Mohammed Parwar from PHU. So these are my collaborators. Anyhow, at the end, I have not done the work. Work is done by my students. 2 students are present over here, and currently I have 6 PhD students. These are my PhD students, and these are the students. They have moved from my lab. They have done the work in my lab. And finally, the funding support. I got the support from UGC, DBT, DST SAR, DST RFPR, recently DST SAR CRG, and ICMR it is approved. We are waiting for fund. Okay. Thank you for your nice attention.
Thanks, Professor Mandal, for your excellent presentation. So, if you have any questions, yes, Subhash.
So it's a nice presentation, Dr. Mandal. Overall, cholesterol is good for cancer cells. This is what you said, right?
Cholesterol is good for cancer cell health.
Cancer cells.
Yes.
So did you also check good cholesterol and bad cholesterol?
No, I checked only total cholesterol. Because inside the cells, whatever I check, it is an intracellular cholesterol. So it is all are bad. It is not HDL or LDL because it is the cholesterol present within the cells. So we don't need to see HDL or LDL when we see the cholesterol within the cells. Okay.
Yeah, Professor Kundu.
Yeah, Dr. Chondi, good. So I have one question.
Yes, sir.
India I have not seen, but outside, many people they have established osteolytic bone metastasis model in rat or mice, especially in rat. Maybe it would be good if you can develop that model and check the efficacy of what you have tested in that in vitro.
Yes, sir. I will try to do it because some problem in actually working in animals. So I have collaboration now in Sanjay Gandhi. They have the mice facility, and hopefully we'll do over there. Yeah. Thank you, sir.
Professor Anil.
Yeah, over here. I'm sorry, I may have missed it, but what is the evidence saying that cholesterol will induce rank-gain?
Cholesterol can reduce the rank ligand.
Yeah, it is.
There is no much evidence, but that is the one we are going to say that when the cholesterol level is more, the rank level is more. But if you keep the cholesterol level, I published the paper in 2017. You can see that when you have treated the cells with simvastatin as well as the MBCD, methyl beta cyclodextrin, that is the cholesterol depleter as well as the cholesterol lowering drug, we have noticed that the rank ligand level as well as the CSF1 level is down regulated.
But just a small comment.
Yes.
The correlation between cholesterol and cancer incidence, I completely agree. But could it be possible that cholesterol itself is not the culprit, but because of the activated cholesterol metabolic pathway, that converts a lot of chemicals that we internalize through.
Yeah, actually, I understand. There are few papers that they are saying the 27 oxycholesterol that could be the potential than the regular cholesterol, but we have noticed that cholesterol can upregulate ARC pathway, ERK1 by 2, that is ARC pathway, and that pathway can be one of the important factors for increasing the, of course, only cholesterol may not do, but one thing that when cholesterol level is more in the cell surface or plasma, that may inhibit the what is called permeability of many molecules and even the oxygen permeability. That may itself cause the hypoxic condition that may increase the cancer potential. That is my hypothesis, even though I have not tested yet.
Dr. Mandal, I have a question.
You work in cholesterol, so why are asking question?
Yeah, because your data and my data don't match at a certain point.
But that's good. Then we collaborate together.
Yeah.
How we can match it?
So exactly. So we had worked on colorectal cancer, and we also did the panel of the cholesterol homeostatic enzymes. And what we found out that HMG-CR level is in fact inversely correlated with the survival. So if you have a higher HMG-CR, the patients are able to cope up better and survive better. Now here is a molecule which is inhibiting HMG-CR, and you are saying that that is the one which is beneficial.
Actually, what happened? Maybe if we analyze again the TCGA database, and we can search, okay? Because there are so many cancers. Maybe colorectal cancer, breast cancer. Maybe
Yeah, we've done that.
But everybody knows that the simvastatin is good for the cancer.
No, there is a controversy about that.
Yeah, there are
Simvastatin. And that's where the personalized medicine comes. And in fact, they talk about this, that the people who have high levels of HMG-CR do not need statins. So So there is a question of personalized medicine
So
because we
in
have
that-
analyzed the data.
Yeah,
from
in that
the-
case, I can tell one.
I will continue that discussion.
I can tell one hypothesis about
We
that.
can discuss during the tea time.
Yeah, okay.
I have a good hypothesis. I'll tell you about that. Why the cholesterol is the simvastatin is not working all the time.
Thank you, Professor Mandal.